Hello, my name is Rene Boiteau, and I'm an assistant professor here at the Department of Chemistry at the University of Minnesota. My research focuses on how metals impact the environment, and today we're going to discuss iron fertilization in the ocean. What is it? Why is it a big deal? And what do we really know about how iron affects marine ecosystems? So usually when iron fertilization makes its way into the news and to international conversation, it's due to a controversy. And so there's a really great example of this that occurred some years ago in the North Pacific Ocean. And it started in 2008 after a volcanic eruption along the Aleutian Islands off of Alaska. So this eruption resulted in a huge amount of volcanic ash that's rich in iron being deposited throughout the North Pacific Ocean. And so you can see in the top here a satellite image of where that ash landed with warmer colors indicating high concentrations of that ash deposited. Right? So later that season, measurements of chlorophyll fluorescent from satellite in the surface ocean, which is a proxy for algae abundance, showed, basically from space, a huge algae bloom all along the track where that ash was deposited. You can see that in these warmer colors in the same swath where the ash landed. If we compare that to previous years when there wasn't a volcanic eruption, we see that this had not been associated with algae blooms. And so there was a hypothesis put forward that iron additions to this region of the ocean created a massive algae bloom. So fisheries along the coast of Canada noticed that in some rivers, they got record salmon stocks in the following years. And one of the hypotheses was that these salmon increases were driven by the fertilization event from the volcanic activity several years before. Right? And so this is very anecdotal evidence connecting potential iron fertilization with fishery output. Nevertheless, regardless of how speculative it was, a company decided to sell fisheries in the region on the idea that they could actually add iron themselves to the ocean to have similar effects on fishery stocks. So in July of 2012, a company was hired by the Fisheries of Canada to dump 100 tons of iron into the North Pacific Ocean. The goal was, again, to fertilize iron that would cause an increase in plant growth, boost the population of salmon, and potentially also sequester carbon in the deep ocean. This project was met with intense criticism from scientists. So the dumping of iron into these international waters, first of all, violates moratoriums on geoengineering and on ocean pollution. It's also not clear that this fertilization would actually have the intended effects. And in fact, it could have had effects that were less desirable, such as harmful algal blooms or actually an increase in certain types of greenhouse gas emissions. Right? The science really is not settled here. Right. And so there's a really big need to understand how iron affects the ocean. And so we can ask in more detail, what is iron fertilization intended to do? So ocean fertilization is the idea that you can add nutrients to the surface ocean to fuel growth of algae that form the base of the food chain. So this was just like if you add fertilizer to your garden with the goal of improving the growth of plants. So if you take a ship out to some region of the ocean and add the nutrient that is currently is depleted and limiting algal growth in that area, you can get an increase in what we call primary production. And the algae that are growing are then taking CO2, carbon dioxide, from the surface ocean, and they're using that to build biomass. So that CO2 that they're taking up is then replenished by more CO2 dissolving from the atmosphere. And this has the effect of drawing down atmospheric greenhouse gas concentrations. So those algae that have grown can then be eaten by higher trophic levels. So there's this potential of that carbon to cascade up to fish and other populations. And then when larger things die, they sink. And ultimately, that takes that carbon that was originally dissolved in the atmosphere and transfers it into deeper layers of the ocean. So the ocean is a stratified system, which means that the surface layer is rarely connected with the deep layer. So deep waters are isolated from the surface with a residence time of about 1,000 years, which means that if you transfer carbon from the atmosphere into those deeper layers, it effectively locks away carbon from the surface atmosphere system. So the goal of iron fertilization is ultimately to increase the growth of these larger organisms and to transfer carbon to the surface ocean, to the deep ocean, to mitigate the effects of fossil fuel burning, which has been increasing CO2 in the atmosphere and resulting in global climate change. So why might iron be a good target for these types of fertilization efforts? Well, it took oceanographers a really long time to recognize that iron was an important nutrient in the ocean. So chemical oceanographers measure the distribution of chemicals across the ocean to understand the processes that are going on. And in the case of looking at biological growth, the most important process is the supply of nutrients. And throughout most of the ocean, primary production is limited by macronutrient availability, especially nitrogen. This is the most abundant nutrient that's incorporated into biomass. And in the early days of ocean sciences, it was thought that organisms would essentially grow until they ran out of nitrate, and that's what would cap primary production. But as we started to map out the concentration of macronutrients across the global ocean, it became clear that in about a third of the ocean, there's really high standing stocks of nitrogen, nitrate, and also phosphorus. So this is shown here in this map, showing nitrate concentrations, cool colors indicating low concentrations, warm colors indicating high concentrations. And so we can see places in the North Atlantic, North Pacific, the equatorial Pacific, and the Southern Ocean have really high standing concentrations of nitrate. And the reason for this had been a mystery for quite a long time. Why were organisms not taking up that nitrate that was just sitting there when there's plenty of sunlight, plenty of water, plenty of carbon dioxide? And so the reason was actually demonstrated through a set of experiments conducted in the late 1980s and early 1990s. And so scientists tested the hypothesis that iron was the limiting nutrient in these regions. And it was the lack of iron that prevented that excess nitrate from being used up. And so scientists went out to the North Pacific and collected water in a trace metal clean fashion. So they made sure that they did not contaminate that water for iron, which had been a major challenge in previous iterations of these types of experiments. And then they added back to that seawater controlled amounts of iron, and they watched what happened over time. And they monitored chlorophyll A, which is a proxy for phytoplankton growth. That's the pigment of photosynthesis. And they also measured nitrate drawdown. And they found that when they didn't add iron, the phytoplankton did not grow and the nitrate did not get significantly drawn down. But as they added small amounts of iron, gradually phytoplankton growth increased and nitrate drawdown increased as well, until if enough iron was added, all of the nitrate could be drawn down. So this was the first evidence that iron was the limiting nutrient in a place in the ocean. And so the scientist who was in charge of these studies, John Martin, presented this at a conference saying, give me a half a tanker of iron and I will give you an ice age. So he had a bit of a flair for the dramatic, but really he was making the connection between these high nutrient regions, which are widespread throughout the ocean, and the possibility that really they're iron limited. And if there are changes in the amount of iron that are delivered to these places in the ocean, you could create massive blooms of algae that would have global implications for CO2 drawdown and for the growth of marine ecosystems. So if enough iron was added, you could actually draw down enough CO2 from the atmosphere, potentially to impact greenhouse gas concentrations to the point where you get climate shifts, cooler climates. Right? So this was a really major dogmatic shift in ocean sciences at the time. The idea was met with a lot of enthusiasm because it started to explain a lot of ocean processes that we struggled to understand previously, and a lot of skepticism as well. So is iron really that widespread of a limiting nutrient throughout the ocean? If it is this actually something that humans should get involved with, right? Should we try to geoengineer the planet to deal with things like climate change by trying to soak up soak up carbon dioxide in the ocean by fertilizing it actively with iron, right? What could go wrong if we try to go down that path? So at this point, it was very clear that we just needed more scientific information to constrain the extent to which iron limitation was an issue in the ocean. And so the following decade was characterized by a series of iron fertilization experiments that tested whether iron was indeed a limiting nutrient in these high nutrient regions across the ocean. So experiments were conducted in the North Pacific, the Equatorial Pacific, and the Southern Ocean, all these major high nutrient regions. And in these experiments, a ship went out and they dumped iron into the ocean. And they measured how much production there was in those patches where there was fertilization and control patches where there wasn't. And in all of these cases, there was a consistent effect on increased primary production in the fertilized regions, right? And so this table is summarizing a lot of the experimental outcomes of these iron fertilization experiments. Primary production increased between 2 and 10-fold due to the addition of iron. And so from this, it was extremely clear that global iron limitation of marine ecosystems is the reality, and changes in the inputs of iron can vastly change marine ecosystems. On the side of ecosystem growth and export of that carbon to the deep ocean, there were much more inconsistent effects. And so in some experiments, about half of the carbon that was fixed by photosynthesis ended up being transferred to the deep ocean. In other cases, it was a much smaller fraction. And it wasn't clear why there were these differences, but it was probably linked to the microbial community that was there at the beginning. So they also saw a very inconsistent effect of what sort of organisms grew after fertilization. And so from this, it was very clear that iron is an important nutrient that controls ocean productivity, but we needed to back up and really understand more fundamentally, how does the iron cycle work naturally? And at this point, we didn't even have a good sense of zero-order questions like how much iron is actually in the ocean. And so in the early 2000s, a group of scientists met and formed a committee that became later the Geotraces program, which tried to assess iron throughout the ocean. And so at that time, there were just very few measurements of dissolved iron throughout the ocean. So this is a map here showing the locations where iron had been measured in 2003 in the interior of the ocean. And so we just didn't really understand where iron naturally comes from. And so internationally, there was a recognition that we needed a program to map out the distribution of iron across the world's ocean to better understand it. So Geotraces ended up launching in later that decade as a collaboration between almost 30 countries to do a coordinated effort of sampling the global ocean and measuring dissolved iron concentrations and other elements in their isotopes and molecular forms with intercalibrated methods that could be used to assess global distributions. And so this program is still ongoing, but as of now, we have a much, much better picture of iron throughout the global ocean. And so this map here is showing an international intermediate data product from Geotraces. And I really love this picture because it's showing dissolved iron concentrations across the Pacific Ocean. And really, for the first time, we're starting to see very clear evidence for where iron is coming from and where it's being removed. So you can see these hot spots in warm colors showing high concentrations of iron along the mid-ocean ridge, indicating volcanic activity is a major source, off of sediments from continental margins. And then we have very low concentrations in the surface ocean where that iron is being taken up by algae that are incorporating that iron into enzymes for photosynthesis and then other organisms that are also using that iron for other biological functions like respiration and nitrogen fixation and a lot of other biological functions. So the other thing that's really striking about this map is that almost everywhere we measure dissolved iron in the ocean, it's actually, the concentrations are far higher than the solubility of inorganic iron, which is only about 8 picomolar. And so the reason for this is that organisms need iron for their metabolism, so they are active at solubilizing that iron. Iron itself is very low concentrations because in oxic seawater, it tends to form rust. It precipitates out as iron oxyhydroxides, right? Unless it's bound to some sort of organic molecule that can keep that iron in solution. And so a lot of the research in my group has been on developing analytical approaches to identify the specific organic molecules that are solubilizing that iron and enabling these higher stocks that end up being distributed throughout the ocean. And there are different types of organic molecules that are produced by different microbes in different places of the ocean that differentially affect how that iron can be taken up. So microbes throughout the ocean can produce organic molecules. Some of those molecules can bind iron very, very tightly, and this becomes this part of the metal complex iron organic pool, right? In the surface ocean, some of that iron can then be taken up by microbes, right? This is what fuels productivity. So it's really important not just to understand the distribution of iron throughout the ocean, but what are the chemical forms of iron? What are generating the organic molecules that keep that iron in solution? And then how available are those forms to the microbial communities that ultimately need to be able to take up that iron to fuel production? And so, as I mentioned, the Geotraces program is still continuing. And one of the most important sections of the ocean that it doesn't appear on this map is the South Pacific, the Pacific sector of the Southern Ocean, this place highlighted in yellow here. And so this part of the ocean is really, really important because the Southern Ocean, as I mentioned, is the largest iron-limited ecosystem on Earth, right? And so delivery of iron to that region is really, really important for fueling carbon uptake by the ocean. And this region of the ocean is also the most susceptible to environmental changes. So one of the things that scientists realized early on is that in the Southern Ocean, if you look by satellite, there are very massive blooms that occur just after sea ice melt events. So in the winter, sea ice forms around the Antarctic continent, and then that melts in the springtime, which is December through February, right? And so at the edge of that sea ice, if we look by satellite data, we can see massive phytoplankton blooms that are occurring in this region. And these blooms are really important, again, for transferring carbon from the surface ocean to the deep ocean. So some of the most intense blooms occur in this region of the Southern Ocean, known as the Amundsen Sea. And this is also one of the regions where sea ice melt rates are increasing the fastest. And so there's a question going forward of, somehow these blooms must be driven by a delivery of iron from somewhere coinciding with the melting of the ice, right? And we want to know in the future, is the delivery of iron going to be more intense or less intense as the dynamics of sea ice melt change, sea ice melt rates increase. And so to understand that, we basically need to understand, where is the iron coming from and what are the important processes involved. And so to that end, the Geotraces GP17 ANT cruise was born to survey the Southern Ocean and the Amundsen Sea shelf to understand where this iron is coming from. And so my research group participated in this program along with a lot of other colleagues from the United States. My student, Nicole Coffey, who's a graduate student here at the University of Minnesota, spent 2 months on an NSF icebreaker, the Nathaniel Palmer, going along the shelf edge and collecting samples across the water column to survey iron and the chemical forms of it all along the shelf during this bloom event in 2024, January 2024. My research assistant, Kate Kuba, was also really important for planning our part of this expedition. And then a postdoc in my lab, Anil Timalsina, analyzed a lot of the samples from this cruise along with Nicole. And so how does this actually work? Well, we collect samples with sophisticated samplers, these rosettes that are a series of bottles that are open tubes with tops and bottoms that can be flapped open when the instrument is deployed. And so on the bottom of this package, there are a set of instruments that can measure the conductivity or the salinity of the water, the temperature, and the depth and chlorophyll fluorescence, which again we use as a proxy for algae growth. We can deploy this and lower it all the way to the bottom of the ocean. And then when it comes back up to the surface, we can trigger the bottles to close at different depths, capturing that water, which can then be sampled on shore. So this is a picture of a video of the deployment, not on the Southern Ocean. You can tell that people are wearing t-shirts, which you wouldn't be doing in Antarctica. But this similar equipment was used on that expedition. Okay, so then we bring those bottles on board. We have makeshift clean labs. We need to be very careful that we're sampling the iron that's present in the water, not iron that's contaminating from our setup. So in the clean room, we filter the water and we fill carboys, and then we bring this into a closed system where we take that seawater and we pump it through solid phase extraction columns that capture the majority of the iron and the organics that they're bound to on solid phase extraction columns. And so this has the effect of pre-concentrating these very, very low concentrations of iron and organic matter, and also removing salts, which would otherwise interfere with our downstream analysis. And then we can take these extracted columns, bring them back to the lab, and then elute them and determine the amount of iron that's bound to different chemical forms. And so the way that we do that is with liquid chromatography coupled with different types of mass spectrometry. So our goal is to separate the different chemical forms of iron, quantify how much iron is bound to each of these different chemical forms, and then also identify the chemical forms to figure out what is producing them and how available they might be to organisms. And so The liquid chromatography that we're using is reverse phase chromatography. So more polar hydrophilic molecules elute earlier, and then more hydrophobic molecules elute later. And we can see that when different samples are analyzed, we have very different chemistries of the molecules that are binding to that iron. So this is showing a chromatogram of retention time. So compounds are eluting over time. And then they're being detected by inductively coupled plasma mass spectrometry, which is essentially counting the amount of iron that's eluting over time in association with different organic molecules. And so we can see this pulse of iron that's associated with hydrophilic organic ligands binding to that iron. That's present mostly in this case in this blue sample, less so in this orange sample. And then eluting later, we have a different suite of molecules that are eluting, and we can start to see chromatographic resolution of several discrete peaks, right? Different molecules that are binding to that iron. And in this case, they're mostly present in those orange, the orange sample, and they're absent from the blue sample. So we can see here that this technique is really useful for differentiating the types of iron that are present throughout the water column and quantifying them independently, right? And then we can identify what those molecules are by coupling the same chromatography with ultra-high resolution electrospray ionization mass spectrometry. So now when the compounds elute from the column, we're detecting the intact molecule, and we can identify those molecules based on the accurate mass of our instrument and also the detection of rare isotopologues. And so we can tell that something is bound to iron because we find different isotopes of iron bound to that molecule, for example, iron 54 and 56, right? We can then find a match to this molecule in databases and then get authentic standards to validate the identity of that molecule. And so what we find is that many of these later eluting molecules are hydrophilic, amphiphilic siderophores. These are molecules that are specifically made by marine microbes in response to iron deficiency for the purpose of mining iron. So they have this peptide-like head group that can bind iron extremely strongly, right? And then this can essentially solubilize iron that's in the ambient environment in a form that can then be taken up directly by the cells through dedicated membrane receptors. And so we can start to separate and differentiate the distribution of different forms of iron across the shelf to understand where are these different chemical forms of iron coming from and what might be the processes that explain that. And so here I'm showing section plots of dissolved iron across the Antarctic shelf with depth. So on the right, we have a map of our sampling station. So the ship came from offshore onto the shelf and then surveyed sample stations along this shallow shelf region. And we can see in this plot, this first plot is looking at only these more hydrophilic iron species, which dominated the total iron concentrations. And we can see that, first of all, these molecules were primarily byproducts of degradation. So they're not metabolites specifically designed to bind iron, but they're being produced by organic matter, by microbes that are degrading organic matter, producing some byproducts that persist that happen to be able to bind iron very strongly. And so from this plot, we can see that these hotspots of these hydrophilic iron forms from right near the sediment interface in certain stations, especially station 23 and station 20. And this is exactly where we have the highest productivity in overlying waters. And so this is giving us some really clear picture that sediments and the sediments, and particularly the sediments where we're producing specific types of organic ligands that can stabilize that iron, are a major source of iron that then fuels production in the surface. And so ongoing work is trying to look at the sediments that were collected in these same regions and try to understand what's unique about them for being able to supply the right types of organic ligands that can then be swept into the surface ocean. The other thing that we recognize is that these amphiphilic siderophores, these more hydrophobic compounds, are primarily produced by microbes that are inhabiting the surface ocean, as well as deeper in the water column in these particular stations where we have a lot of iron coming out. And so what this represents is the fact that these degraded organic iron forms are not necessarily directly available to organisms. They can't take up these degradation products, right? And so what they do is they produce organic molecules that can rip the iron away from those products and other particles that might be sinking through the water column. And then that chemical form is something that they can take up through dedicated membrane receptors. And so it shows, the specific strategies that marine microbes are using to mine that iron. And we can, from knowing the molecules, we can associate the production of those mining molecules to the abundance of certain types of bacteria that need to be present in the water column to make that iron bioavailable. And so the upshot of this is that what we're starting to understand is that iron availability in the Southern Ocean depends on the sediment chemistry. So how much of this organically bound iron is being produced by specific sediments. The ocean physics, so that iron has to get from the deep ocean all the way to the surface. And of course, the ocean physics can change seasonally, and it's likely driven by these melt events. So that explains why the timing of delivery coincides with sea ice melt rate, sea ice melting. And then finally, it likely also depends on the bacterial community. So you need this handoff from the iron bound to these degraded molecules, and to prevent it from just sinking, absorbing to some particle and sinking out, microbes have to produce molecules that will rip it away and then facilitate its uptake. And so this gives us a picture of the different processes that are important for supplying iron to the surface of the ocean. And ongoing work is trying to really put this in the framework of models that can then predict how these processes are likely going to change in the future. And so, moving even further into the future, there really needs to be more additional studies on longer term effects of iron fertilization. And so there will, the idea of actively fertilizing the ocean with iron as a human geoengineering strategy that could sequester carbon or influence fisheries was largely shelved after the initial iron fertilization experiments were conducted, because there was viewed that it was just too risky to do something like that. Well, decade or so later, we're in a scenario where atmospheric CO2 concentrations have continued to increase at very, very high rates. And so there's worry now that, the worst case climate scenarios could be very, very bad. And we need to start thinking about ways to actively remove carbon from the atmosphere. So National Academy's report coming out in 2021 made a call to have additional research on iron fertilization as one of the greatest potentially impactful strategies for ocean carbon dioxide removal, right? So we really need to address this question when iron fertilization occurs, under what scenarios can it have the desired effect of sequestering CO2 or increasing fishery stocks? And on what cases could you have deleterious effects, things like fueling harmful algal blooms or producing nitrous oxide or causing oxygen deficiency, which are all other types of outcomes that have been observed in some cases linked to iron fertilization. And so to summarize, ocean iron fertilization, whether we intervene or not, is a really important process that influences global ocean productivity. Right? So because iron has a very short lifetime in the ocean, only about 1 to 2 years, the sources and distribution of iron in the ocean can change very rapidly on the timescale of human activities. And so in 10 years, right, the supply of iron to the ocean might look very different, and that can change how much productivity there is going to be in the global ocean. So it's really important to understand what these and predict what these effects are going to be. In the Southern Ocean, we're starting to see that changes in ocean circulation patterns are likely going to be a major driver of the extent to which naturally occurring iron is fertilizing the surface, right? So certain sediments seem to be a very important source, and whether or not that water gets advected to the surface is going to be a major control on the delivery rate and magnitude of iron. Going forward, there's a need to better understand factors that are modulating the ocean's responses to iron fertilization, as well as what are the longer term effects, right? So can we start to track the effects of iron fertilization on things like fisheries, which may have 1 or 2 years of delay between the time of a fertilization event and the observed outcome. So with that, I'd love to thank my research group, especially Nicole Coffey, Anil Timolsina, and Kate Kuba, who participated in this cruise and generated the data, as well as collaborators on the Geotraces GP17A and T project, especially Dan Rapetta, Jay Lee, Yulia Strenga, Phoebe Lamb, and Pete Sedgwick, and the other scientists and crew on the expedition. And this project was funded by the National Science Foundation, as well as the Sediments Foundation. I thank you for your interest in this topic.